Okay, this is lecture 1, part 2. We're talking about blood. We're looking at we ended part 1 with talking about the signals that trigger production of red blood cells. And this slide here is a overview of the full life cycle of a red blood cell. So depending upon oxygen levels, the kidneys will produce more or less erythropoietin, and with a low more or less of the hormone erythropoietin. With lower levels of oxygen, measured as the partial pressure of oxygen, kidneys produce an increase in erythropoietin. In any case, the erythropoietin is a hormone that's secreted, as all hormones are, into the bloodstream, and it goes to the red bone marrow, where it triggers production of erythrocytes from the primitive hemocytoblast stem cell. And those erythrocytes have a lifespan of about 100 to 120 days. As they get older and they age, there's changes in the surface membrane which mark them for engulfment phagocytosis by macrophages, mostly in the liver. And then they're broken down, and the different parts of the molecules of hemoglobin, which comprise most of the protein inside the red cells, all the components are recycled. The protein component, globin, gets broken down to amino acids that can go out into the bloodstream and get taken up by other cells to make other proteins. Heme is further broken down into bilirubin, and the iron is stored separately. Iron in the body is always bound to something, because free iron itself is toxic. So it's always bound to some sort of carrier molecule. The bilirubin is secreted into the intestine in the bile, and is involved in what we'll talk about later in the course. It's involved in uptake of fat from the food. Part of it also is broken down by bacteria to form a molecule called stercobilin, which is brown, and that's why feces are brown, because of stercobilin produced by bacteria from bilirubin. Bilirubin is also reabsorbed in the intestine, so you have this hepatointestinal recirculation. Okay. There are many disorders. Some of these are overlapping. If abnormally low oxygen carrying capacity in the blood is defined as is the definition for anemia, but it's a symptom rather than a disease. It can be the manifestation of many different diseases, or something as simple as blood loss due to bleeding. Whatever the case is, anemia is associated with inadequate oxygen levels in the blood. The signs for that include fatigue, paleness, shortness of breath, chills. Anemia can be due to insufficient erythrocytes. There's a number of reasons why a person could be anemic, but insufficient erythrocytes, for example, hemorrhagic anemia resulting from either a very quick, large loss of blood, an acute loss of blood, or a chronic, slow bleeding that continues for a long time. Either one can, and most of these chronic blood losses are actually internal rather than external. That can cause anemia, which is, this would be a hemorrhagic anemia. Hemolytic anemia involves the premature lysis or rupture of the red cells. And aplastic anemia is destruction or inhibition of the red bone marrow, so you don't have you have a lack of adequate production of cells from the bone marrow itself. They never get produced. So hemorrhagic, hemolytic, or aplastic anemias all result in insufficient red blood cell numbers. Sometimes anemia is due to a decrease in the hemoglobin content inside the cells. It could be due to iron deficiency anemia. Iron deficiency anemia can be a result of hemorrhagic anemia, because with excess bleeding you have loss of iron, or due to inadequate uptake of iron-containing foods, ingestion of iron-containing foods, or sometimes it can be due to some sort of problem with iron absorption. Another subgroup of anemia resulting in decreased hemoglobin content would be pernicious anemia, which is a result of deficiency of the vitamin B12. Vitamin B12 is absorbed from the gastrointestinal tract only with presence of a molecule called intrinsic factor. Intrinsic factor is produced in the stomach into the gastrointestinal tract, and it's needed for absorption of B12. Treatment for this kind of anemia, pernicious anemia, is simply intramuscular injection of B12. There are other genetically based anemias due to some abnormal genes which code for abnormal hemoglobin molecules. Thalassemia is a genetically based disease which results in the absence or of a globin chain, or a faulty globin chain, a change in the globin chain so it's not properly functional. Another type of genetically based disease resulting in abnormal hemoglobin is sickle cell anemia. In this case, the defective gene codes for an abnormal hemoglobin, which is called hemoglobin S. These kind of people with this disease, and they have to be homozygous for the gene, will experience a change in the shape of red blood cells when the PO2 drops. Now, you have lower PO2 when you're exercising a lot, or going to a higher elevation, or if you have some problem with a lung function, so you can't take up oxygen as well. And so you get a low PO2 environment inside the body, and that results in a change of hemoglobin molecules inside the cell, so that the entire cell changes its shape into this kind of an arc shape. It is similar, this kind of arc shape, which is similar to a type of knife called a sickle. The problem here is that these sickle-shaped cells, just when a person needs more oxygen, the low oxygen environment inside results in this change in shape, and these sickle-shaped red cells have difficulty passing through capillaries. Now, another type of abnormality is called polycythemia. Here, it's a question of having excess red blood cells. The problem with that is that it increases the thickness or viscosity of the blood. That's a problem. There are different types of polycythemia. A genetically based type of polycythemia is called polycythemia vera, where you have abnormally high red blood cell production. That's not necessarily at all difficult to treat, because what they do is they remove on a regular basis some of the person's red cells, and they function extremely well. Secondary polycythemia is a type of polycythemia that is it's a secondary effect of some other process. One type of secondary polycythemia is due to what's called blood doping. Blood doping involves blood doping involves either transfusion of excess amounts of blood into a person or injection of erythropoietin into a person when they don't need it, when their hematocrit, their red cell levels are fine, or it can be due to being chronically in a low partial pressure of oxygen environment. So if a person, let's talk about this one first, if a person goes to a higher altitude, with time, it takes quite a few days, it takes a couple of weeks at least to trigger the erythropoietin and the increased production of red cells. But if at a higher altitude, the PO2 in the atmosphere is lower, and therefore that will cause a hypoxia in the tissues, and the kidneys will produce more erythropoietin. So that can trigger a polycythemia secondary to that low atmospheric oxygen levels. Some athletes have in the past injected erythropoietin to trigger more red cell production, because that will enhance their ability to perform in athletic competition. The increased oxygen carrying capacity allows them to have increased ability to produce ATP, energy source from aerobic respiration. This is a detectable kind of blood doping or cheating. It's considered to be cheating, and it's detectable because the erythropoietin is synthetic type of erythropoietin. Well, synthetic. It's produced in the lab because the gene that codes for the molecule erythropoietin, the protein molecule erythropoietin, has been cloned and put into cells. So it is available as a recombinant protein, and is used for many patients who need more erythropoietin. They're injected with it. Very common, quite commonly used for treatment of people. And so there is a black market, but it is fairly easily detectable in the lab, so that doesn't work out. Another type of cheating or blood doping is some athletes would in the past have had some of their blood collected, like a bag of blood, a unit of blood collected 2-3 weeks before competition, and then their body responds by producing more blood to bring them back to the normal level, and then just before the competition it's reinfused into them, so it's an auto transfusion. So what happens, of course, is they have a higher hematocrit, a higher hemoglobin content in their blood, and they can again carry more oxygen. Their oxygen carrying capacity rises. This also is relatively easily detectable because the blood collected has to have an anticoagulant in it, and so they look for a presence of anticoagulant. And presence of anticoagulant is illegal to have in your blood. So actually, for athletic competition, the only legal thing is to move to a low PO2 environment. Now, an expensive way of doing that is to go to somewhere like Denver, Colorado, for example, and train there. A cheaper way, which is not that uncommon, not that uncommonly used by athletes, is to sleep inside a airtight tent that has an artificially low PO2 environment. And that's enough if they sleep every night for weeks in that kind of environment. That will trigger their kidneys to produce more erythropoietin, and it's perfectly legal in terms of athletic competitions. So blood doping is kind of an interesting topic, actually, as I would say. So that's it. There are cells in your blood, including red cells, and white cells and platelets, which are subcellular fragments. And this is the breakdown of the white blood cells, which we're going to talk about now. The white blood cells are the only cells that have a nucleus in the blood, much less numerous than the red cells. They can leave the blood capillaries. Only the capillaries, everywhere else all the blood vessels, all the other blood vessels other than capillaries, are simply for transport, either transporting oxygenated blood to the tissues or bringing partially depleted, oxygen depleted blood back to the heart. All exchange occurs in the capillaries. These are very, very thin, tiny blood vessels where gas exchange, nutrient exchange, and metabolic waste exchange occurs. And also white blood cells can leave, if signals or conditions are correct, can leave the vasculature, the vascular space, and move into the tissues, and that process is called diapedesis. When white cells move out between the cells that form the wall of the capillary. When we look at white blood cells, typically you look at the distribution of the different types of cells, whether it's a normal distribution or abnormal, and that's done by doing a differential count. You have to differentiate between the different types of white blood cells to get an accurate picture. This can be done actually with automatically done with a machine, a cell counter, and a machine that identifies the different types of white cells. One thing that is a very clear sign of some sort of infection process, even before the person has any symptoms, you often can see an increase in white blood cell count, total white blood cell count, which is called leukocytosis. It typically precedes symptoms. It's a sign that there is some sort of infection process going on. So again, here you see a photomicrograph of a smear of blood with lots of red blood cells and some white cells. The granular cell, the granulocytes, are also called polymorphonuclear cells because they have multi-shaped nucleus. Poly meaning multi, morpho meaning shape, and nuclear referring to the nucleus. And as a acronym for that, they're called PMNs. You might also hear them being called polys. There are three types which differ from each other by the type of granulocyte or granules found in the cytoplasm: neutrophils, eosinophils, and basophils. Here you see a neutrophil, which has essentially the multi-lobed nucleus, the polymorphonuclear shape of the nucleus, and the different lobes of the nucleus are held together by small. It's all continuous. It's one nucleus, but it's held together by these thin strands between the different lobes. Difficult to see in this photomicrograph. It's actually quite clear. I'll get rid of these arrows now, and you'll see it. So that is a neutrophil. It has neutral staining granules. This is a basophil, which has dark blue or violet, purple staining granules. You can see the nucleus. It's actually a different type of purple and not as granular as the granules in the cytoplasm. This is a eosinophil. It has red, often even more red than this, red granules, often two lobes to the nucleus, but sometimes more. All are phagocytic, but actually its most phagocytic activity is in the neutrophils. They are certainly the most phagocytic cell. These are relatively short-lived cells, much shorter lived than red blood cells, which I remind you have a 100 to 120 day lifespan. The neutrophils account for 60% plus or minus 60 plus or minus 10% of the white cells. There are different types of granules. In neutrophils, you have the granules contain different types of enzymes and proteins, all involved in either directly or indirectly in killing bacteria or fungi. They either are enzymes that can produce toxic compounds, they are either enzymes like peroxidase that can produce toxic compounds, or they are enzymes that can directly hydrolyze by hydrolysis the components of bacteria that are phagocytosed, or they are proteins like defensins that can directly kill bacteria. So these are one of the two major phagocytic cells in the body. The other major phagocytic cells in the body are monocyte macrophages. So the neutrophils are involved in inflammatory responses. You'll get an accumulation. If something foreign goes into the tissues, you'll get quite a rapid accumulation of neutrophils at the site, trying to engulf, phagocytose, and kill, if there is any bacteria in whatever penetrated the tissue. Eosinophils are a fairly small number in terms of the their overall percentage of the white cells. They have red staining granules, often are bilobed, but not always, and they are typically involved in responses or defense mechanisms against parasitic worm infections. A large percentage of the world's population, I would say the majority of the world's population, have parasitic worm infection, are infected with parasitic worms. It's very, very, very common. We here also have a significant percentage of the population carrying parasitic worms, but because our water system is carefully monitored and clean, and the food is regulated, food production and sales are regulated, it's less so than in other parts of the world. These are nice photomicrographs of eosinophils. Here you see a typical eosinophil. I've put this one in because it has 3 lobes actually to the nucleus, showing that it's not always 2. Basophils account for an even smaller percentage of the white cells. I'm not going to ask you the exact number here. It's enough that it's small. It has several multi-lobed, just like any other granulocyte, multi-lobed nucleus. When a basophil is found in the bloodstream, when it leaves the bloodstream and goes into the tissues outside of the bloodstream, it's called a mast cell. Mast cells and basophils contain purple staining granules, and those granules, among other things, also are there's a lot of histamine in the granules, and histamine is an inflammatory compound. It causes vasodilation, relaxation of the blood vessels, so it enlarges. And if the blood vessel diameter enlarges, then you can get more flow into that area. It also triggers an increase in the permeability of those of the vasculature. So wherever you have local histamine release, you get not only more flow because the diameter of the capillaries widen, but also the ability of both fluid and even cells to get out of the vasculature increases because of the increased vascular permeability. And it acts as a chemoattractant for other white blood cells. So it's quite a handy type of molecule to have present to trigger inflammatory reactions that can help in defense against infection. And here you see a photomicrograph of a basophil. The granules are so dark that it's really almost impossible really to see any of the nucleus. Here you can just make it out, but ordinarily it's just, full of dark purple granular cells, and it's all purple inside. Now, I've used the term here non-granular because I wanted to get away from this term that your book uses, which is agranulocytes. These are cells that don't have a clear granules in their cytoplasm, although actually monocytes do have some granules in the cytoplasm. It's just not very clear. They can't be seen. These type of cells, monocytes in the bloodstream, have a kidney-shaped or U-shaped kidney, mostly really a kidney-shaped nucleus. They are highly phagocytic. They're the other main type of phagocytic cell in the body after neutrophils. They're very phagocytic. When they're in the bloodstream, they're called monocytes. As soon as they move into the tissues, they are called macrophages. So you find macrophages in the tissue, having left the bloodstream. While in the bloodstream, they're monocytes. And quite often you'll see these cells referred to, and I'll refer to them quite often as monocyte slash macrophages. So monocyte macrophages. Very commonly found in all kinds of tissues, and the bloodstream as monocytes, and in all kinds of tissues as macrophages. They are 4 to 8% roughly of the white blood cells. Large cells, relatively large cells. Here you see this indented or kidney-shaped nucleus on a monocyte. They can leave the circulation and enter into tissues and then differentiate into macrophages. These are somewhat larger, but they retain their nuclear characteristic indented or kidney-shaped nucleus. Macrophage monocytes, or monocyte macrophages, is a particular lineage produced, of course, it's a blood cell, so it's produced in the red bone marrow, but from the primitive stem cells or hemocytoblasts. All the cells that are mentioned below are part of this lineage of macrophage cells. Very mobile, very actively phagocytic. They're involved actually in activation of the lymphocytes, white blood cells. When the white cells mount a specific immune response, there is a role for macrophages, and if we have time, we'll discuss that when we talk about immunity and the immune response. Now, monocytes, there are different kinds of monocytes, or rather monocyte-derived macrophages in the tissues. Monocytes leave the bloodstream and populate different tissues throughout the body, and they become what are called resident macrophages. They sit there and wait for something to come along that's a foreign object or foreign microorganism that they would attack and phagocytose and try and destroy. So almost all of your tissues actually have this resident bone marrow derived macrophage populations. In the liver, there are Kupffer cells. In the skin, there are dendritic cells. In the lungs, there are alveolar macrophages. In the brain, there are cells called microglia. And in the peritoneal cavity, there are peritoneal macrophages. Those are just a few of the different populations in different areas: liver, skin, lungs, brain, abdominal cavity, peritoneal macrophages in the abdominal cavity. All of these cells are there waiting to act if need be. The other kind of macrophages you have are the ones that are responding to inflammatory signals that are there because monocytes moved out of the bloodstream into the tissues due to an inflammatory trigger, like something foreign getting into your body. And these are called inflammatory macrophages. They're part of the inflammatory response. So these cells are there and present and resident, and these cells got there because they move quickly into an area due to some sort of trigger signal, something foreign getting into your tissues. All these different cells are macrophage lineage cells. The other type of lineage of cells are the lymphocytes. These lack any visible granules in their cytoplasm. They have quite a round nucleus. There are two major populations. There are many subpopulations to each of these, but there are two main populations. There are T lymphocytes, also called T cells, and B lymphocytes, also called B cells. And probably when I talk about them, I'll call them T cells or B cells. These lymphocytes, both T and B cells, form what's called the adaptive immune system. It is a specific immune response. They can mediate the very specific immune response. Each different unique T cell or B cell produced in the bone marrow will have a unique target that it can react to. The lymphocytes, it's not particularly round, but a reasonably round nucleus here and not too much cytoplasm. They account for 25% of the white cells. Therefore, in order of count or frequency, the most common are neutrophils, second are lymphocytes, third would be basophils, I guess, and then eosinophils are the other way around. Both populations are quite small. They are in the bloodstream. Lymphocytes are in the bloodstream, and they're also found in lymphoid tissues like lymph nodes and the spleen. You have many lymphoid tissues in solid tissue in different parts of your body, and some are also circulating in the blood. As I said, the two main types are T cells and B cells. T cells are involved in cell-mediated immunity, and B cells are involved, specific, by the way, immunity, and B cells are involved in antibody-mediated specific immunity. So the B cells secrete antibody, but the T cells and the antibody can interact with foreign molecules. But the T cells don't secrete the molecule that recognizes a foreign substance. They have the receptor on the surface and they keep it so it's cell-mediated. So as a summary here, which one was more common, the basophils or the eosinophils? I think it's the eosinophils are more common than basophils. So here are some general terms, and you can see the lifespan. Red cells lifespan 100 to 120 days. Neutrophils, it's very short, probably maximum is 2-3 days maybe at the most. Eosinophils is longer. The white blood cells, the lymphocytes have quite a lengthy, sorry, that's monocytes. So lymphocytes can be anywhere from hours to years of lifespan. It can be long lifespan, sometimes for your entire life. Monocyte macrophages can be a long time. And then one lineage that we haven't spoken about is one lineage of cells we haven't spoken about are platelets, and they're fairly short lifespan. So production of each of these types of cells is white blood cells is called leukopoiesis, just as production of red cells was called erythropoiesis. And again, it's hormonally stimulated by soluble protein molecules, many different types of soluble protein molecules that are called cytokines. These are proteins or hormones, protein hormones that support, trigger blood cell production, hematopoiesis, specifically white blood cell production, which is leukopoiesis. Some of these cytokines, these soluble signals, are called interleukins, and there's many. They're numbered. IL-1, 2, 3, 4. It goes on quite high. Or some are called colony stimulating factors, or CSF. One of the major, although not only, source of cytokine production are macrophages and T cells, or T lymphocytes. These hematopoietic signals, hormones, or factors are also, many of them being cloned and are produced as recombinant proteins and are used clinically to help stimulate bone marrow production. An example of that would be granulocyte macrophage colony stimulating factor, which helps support the granulocyte monocyte lineage of cell production of those cells. Here you see white blood cell lineages. I want to point out to you that the stem cell is the hemocytoblast. What isn't shown here is, but as you already know, that there is an erythroid line, which if this cell gets committed to red cell production, it will go along the erythroid line, leading to red blood cell production. There is also a megakaryocyte line leading towards megakaryocytes and platelets. I shouldn't draw it maybe this way, just a second. So there is a erythroid lineage, or red cell lineage, and a megakaryocyte or platelet lineage. And here we're looking at the leukocyte lineages. On the diagram itself, it's showing the leukocyte or white blood cell lineages. It's an S, supposedly. So, in terms of lineages, there are two major ones here: the myeloid lineage and the lymphoid lineage. The lymphoid lineage leads to T cells and B cells. I would ignore this for now, but T cell and B cell production. And the myeloid lineage has two further splits, one leading to granulocyte production. Here, the lineage that leads to granulocytes, eosinophils, basophils, and neutrophils, and the other leading to monocyte macrophages. So we have hemocytoblasts going to erythroid lineage, megakaryocyte, myeloid that has two subdivisions, one for granulocytes and the other for lymphocytes, and the other for monocyte macrophages, and then the lymphoid lineage leading to lymphocytes. So the cells in the stem cells in your bone marrow can go in different directions in terms of what's going to get produced, depending upon what kind of hormone signals they're getting from hematopoietic factors, cytokines, and colony stimulating factor that are released in response to usually the need for more of one or the other type of cell. It's a dynamic situation, and it's a delicate balance between the different lineages, and it's an ongoing continuous process because these cells do not live forever, so they have to be replaced. And here you see the different lineages: the megakaryocyte, erythroid, myeloid, and lymphoid, and what they lead to: platelets, red cells, granulocytes, and monocyte macrophages, and here for lymphocytes. They all originate from hemocytoblasts, which are pluripotent stem cells. Pluripotent means that they can go to in different directions depending upon the signals they get. They can differentiate into different cell lineages. Two white blood cell lineages are myeloid and lymphoid. As I said earlier, I wouldn't worry about these other terms. The myeloid, stem cell comes after the hemocytoblast, and there's different stages. But in the end, you either have eosinophils, neutrophils, and basophils, granulocytes, or the other part of the myeloid lineage are monocytes. And the lymphoid stem cells, or the hemocytoblast going to a lymphoid, committed to a lymphoid line, will develop into lymphocytes, T cells, and B cells. And again, here you see the lineages, at least for the white blood cells, or leukocytes, leading to the mature cells here and here. And there are disorders in white blood cell production, particularly tumors of the white blood cells, which are called leukemias. It's a huge topic and cannot be, really discussed in one slide. It's ridiculous. You can do 1,000 slides and not cover all that's known. But leukemia is a tumor derived from white blood cells. And the many different types of leukemia, they're named according to the white blood cell that's involved and that has led to the abnormal proliferation. So if it's myeloid, then in nature it's often you'll have myeloblasts. If it's lymphoid, you have lymphocytes. I mean, it really depends on which type of, and often you'll get a primitive looking cell, poorly differentiated cell. There are acute forms of leukemia and there are chronic forms. The acute forms typically involve less well differentiated cell types and are primarily occurring in children. And then the chronic leukemias, which are more common in older people. There are many good treatments for acute leukemias, and many childhood leukemias have a high success rate in treatment. Chronic leukemias less so. What you have in a leukemia, as I was trying to explain earlier, but it's maybe said better here, you have immature abnormal, that's an important part that's missing here. Immature and abnormal white blood cells are found in the bloodstream and also in the bone marrow. The bone marrow can become totally occupied with these or taken over with these because there's so much growth of the cells, immature abnormal cells. Death often is caused by internal hemorrhage or overwhelming infections because they don't have proper platelet numbers and they don't have proper immunity, white blood cell function. Treatments for leukemias, there are many. Irradiation is used. Anti-leukemic drugs, which people tend to call chemotherapeutic drugs or chemotherapy. But, truth be told, any compound, chemical compound put in is a chemotherapeutic drug, including, by definition, even antibiotics are chemotherapeutic drugs. So, anti-leukemic drugs, which most people call chemotherapeutic drugs. Bone marrow transplants are used, preceded by irradiation, and then bone marrow transplant from as close related donor or matched donor as possible. And then, and I should have put it below, just because it's the most recent development, checkpoint inhibitor monoclonal antibody therapy. This is a tremendously innovative, effective, and low toxicity related. Has a low level of toxicity associated with it. Monoclonal antibodies that act on what are called checkpoint inhibitors, and they allow the immune system to proceed and kill the person's own immune system to go ahead and kill the tumor. And they're quite an amazing new development. And in fact, the initial developers won the Nobel Prize, I don't know how many years ago now, 4 or 5 years ago. Okay, checkpoint inhibitor antibody therapy, very good, really good. First really good use of antibody for immunotherapy, really good immunotherapy. And that's it. That's the end of Unit 1, Part 2.